Indirect method is basically a way to package the gene into the cells of the person that is supposed to receive the gene therapy. In order to do so, we need to extract a portion of the cells from the person and then grow them in a lab, hence the term in vitro. So the process basically involves harvesting a bunch of cells from a person, and then we infect those cells with our vector that contains the gene. And then we monitor the expression of that gene, and after some time, when we have a substantial amount of product inside those cells, we transfer the cells back into the patient. Now, why do we do this? This is a laborious process, but we have to go through it because it minimizes the chance for rejection as well as the chance for triggering an immune response. And now it's time to talk about the molecules that we've been constantly referring to. So let's talk about vectors. Vectors are molecules that are used to transfer your desired gene into the human body. Now that transfer could be directly or indirectly. And as you can see, that generally carrier molecules can be categorized in 4 ways. However, you need to remember that vectors are broadly divided into 2 main types. Vectors can be based on viruses, or they can be non-viral particles. There's a question that I would like for you to think about. With the recent pandemic, we've come to an understanding about viruses, and if you really don't know anything about viruses, I would really like you all to visit Professor Vincent Racanello's channel on YouTube. He teaches about viruses, and he's also the author of the virology textbook that most universities commonly refer to. We can understand particles that are not viruses to transport our desired gene. But I want you to think about why using viruses to transport our particular gene of interest when we know that these viruses can cause human disease. In the next video, we are going to look at certain viruses that are commonly used for gene therapy.